All right, this is Unit 2 where we're going to talk about microbial cell biology, cell structure and function, and cellular activities. Now, a lot of this, what I'm going to discuss with you today, is not in your textbook. Instead, I have kind of pulled out some information from these textbooks here and also ASM and CDC. All right, so let's talk a little bit about the evolution of microbiology. So bacteria and protozoans, they have been observed microscopically for over 300 years. They were first observed by a Dutch biologist known as Anton van Leeuwenhoek, who he called the bacteria that he saw microscopically the wee beasties. You've probably heard of that before. Microbes are found in all types of environments. Not all microbes are going to cause damage, but many will. And some microbes are considered to be normal flora and are actually beneficial to the host, and other ones are pathogens. But the normal flora that is beneficial to the host, they do all kinds of things, including breaking down bilirubin to urobilinogen. They synthesize vitamin K. They compete with pathogens for nutrients and minerals. Okay, the activities of bacteria play a large part on the environment and also play a large part in the balance of life. Without bacteria, we would have some major problems in the world. We need bacteria. It is actually essential for the nitrogen, carbon, and sulfur cycles. Each microbe has its own unique physiology and metabolic pathways that allows it to survive in a particular habitat. The pioneers of microbiology that I want to talk about today are Anton van Leeuwenhoek, Louis Pasteur, and Robert Koch. So Anton van Leeuwenhoek is a Dutch biologist. He was the father of microbiology and observed his wee beasties in the late 1600s microscopically. He was also a lens maker. He was also a biologist. Louis Pasteur was a French chemist that lived in the mid-1800s. He was a professor of chemistry and developed what's known as the germ theory of fermentation, and he also discovered pasteurization, which is the process of heating that kills organisms. And then finally we have Robert Koch, who is a German physician. He lived in the late 1800s and was instrumental as a bacteriologist for his discoveries with anthrax and tuberculosis organisms. He developed or came up with Koch's postulates. So you might be asking yourself, what are the main roles of the clinical microbiologist? And again, you want to isolate, identify, and analyze. So you want to isolate cultures purely, identify organisms using fermentation and metabolic activity knowledge, and then we want to analyze but determine sensitivity or resistance to certain organisms, and also determine if the organisms we isolate are normal flora or pathogens. So let's go ahead and go back here to Koch's postulates. Again, Robert Koch was a German physician, and in 1878, Robert Koch infected 6 animals with 6 different organisms, then cultured those animals and came up with his Koch's postulates, because those 6 animals that he inoculated with an organism ended up growing out those organisms purely. Okay, so Robert Koch did some work with cholera and typhoid fever. But let's talk about Koch's postulates. A given organism must be present in every case of a given infectious disease. So a specific organism is always associated with a given disease. The microorganism can be isolated from a specimen, and that specimen is associated with a disease state. So the specific organism can be isolated from a diseased animal and then grown in pure cultures. Okay, the cultured organism will cause disease if put into a healthy animal. The same organism that is associated with the disease state can be recovered from representative specimens from the experimentally infected animal. So the same exact organism can be cultured from the newly infected animal. Okay? So you might be asking yourself, why do we care about microbial structure and physiology? Well, we have to have an understanding of prokaryotic cell structure in order to identify bacteria by phenotypic, these are traits that are outwardly expressed, genotypic or metabolic measures. Knowledge of microbial structure and physiology is important in the growing of human pathogens, the identification of human pathogens, and finally in the treatment of infections caused by human pathogens. And if you're wondering what a pathogen is any agent that causes an infection. There are many subfields within the field of microbiology, and some of the subfields would be bacteriology, parasitology, mycology, and virology. Bacteriology is the study of bacteria or the unicellular prokaryotes. An example of a bacteria would be something like Staphylococcus aureus. Parasitology is the study of parasites, and these are eukaryotes. They are unicellular or multicellular worms. Okay, some examples of parasites would be things like pinworms, also known as Enterobius vermicularis, and Giardia lamblia. Those are some examples of parasites that you may have heard of before. Mycology is the study of fungi, eukaryotes that obtain nutrients through absorption. Some examples of fungi that you may have heard of would be things like penicillium and also Stachybotrys. That's the black mold that some people have in their houses that cause all kinds of respiratory problems. And then finally we have virology, which is the study of viruses. This is a cellular infectious particle that contains DNA or RNA. Now some of the DNA viruses would be herpes or hepatitis B, and some RNA viruses would be hepatitis A, hepatitis C, and West Nile virus. So those are the four fields of microbiology: bacteriology, parasitology, mycology, and virology. Let's go ahead and talk about classification, taxonomy, and nomenclature. And this is basically naming the bugs or naming the organisms, okay? So taxonomy is grouping organisms into categories. It refers to three concepts: classification, nomenclature, and identification. Based on the genetic makeup of an organism and the observable physical and functional characteristics of that organism, that is how we come up with their name. Now, classification and nomenclature are part of taxonomy, or nomenclature and classification together is taxonomy. Classification is dividing organisms into groups. Organisms are classified by genotypic or phenotypic characteristics as well as by cellular type. Okay, and then nomenclature is basically naming the bugs or naming the organisms and how they are written. So bacteria, for example, when you spell that out, Staphylococcus aureus, either has to be italicized or it has to be underlined. So the genus name of the organism is always capitalized, and the species name follows the genus name but is not capitalized. Both are underlined or italicized. So a good example of taxonomic classification is the example of Escherichia coli. This was named after Theodore Escherichia, who isolated bacterial cells from infant feces in 1885. E. coli is an organism common and normal in the gastrointestinal tract. Now, there are some strains that can be pathogens, but most of the E. coli that lives in your gastrointestinal tract is normal. So the name Escherichia coli comes from Escherichia, taken for Theodore Escherich, and coli, which is Latin for of the colon. Okay, there are a lot of organisms that have a species name of coli, such as Entamoeba coli, Escherichia coli, and Balantidium coli. So let's take a look at the taxonomy a little bit more. For E. coli, the domain is bacteria, the kingdom is bacteria, the phylum is Proteobacteria, the class is Gamma Proteobacteria, the order is Enterobacterialis, and the family is Enterobacteriaceae. Okay, anytime you see something ending in ACA, that refers to the family. And then the genus, you can see there is italicized and capitalized, is Escherichia, and then the species is coli. Now there can even be certain strains or tribes, like a strain could be E. coli 0157H7, and then they can also be broken down into tribes, and we'll talk more about that when we get to the enteric group. So that's the taxonomic classification of E. coli. The prokaryotes are your bacteria, and the eukaryotic cells are things like us. We are eukaryotes, and multicellular worms are eukaryotes. So let's compare the prokaryotes to the eukaryotes, okay? The prokaryotes, these are usually very small organisms, usually 1 to 10 micrometers in length. They have no mitochondria. Mitochondria makes energy to power the cell, so they have no mitochondria. They have no endoplasmic reticulum. That is a structure that transports proteins, and they have no nuclear membrane. They have only a condensed DNA molecule, and typically they're very small. Like I said, 1 to 10 micrometers, but usually they're less than 5 microns. Okay. Externally, the prokaryotes will have things like flagella for motility or pili for adhesion. And if they have a capsule around them, that's called glycocalyx. That is a slime layer around some of the bacteria. Okay. So the prokaryotes have no nuclear membrane. They are made of a phospholipid and protein structure, but no sterols. Now, prokaryotes lack a nucleus and membrane organelles. They are very different than eukaryotes because they have their internal structure, because of how their internal structure is organized. Okay, a eukaryote has a mitochondria, endoplasmic reticulum, and Golgi bodies. And a Golgi body is something that sorts and processes proteins in a cell. Okay. They also have a classic membrane-bound nucleus, and usually they're larger at more than 10 microns. Eukaryotes can have cilia, flagella, and pili also. Okay. Let's go ahead and compare a little bit more of the prokaryotes and the eukaryotes in terms of cell structure. The prokaryotic cell structures would include things like cytoplasmic structures, chromatin body, single chromosome. Okay, a chromatin is basically genetic material, and they have a single chromosome. They have ribosomes, which assemble proteins, cytoplasmic granules, and endospores. And an endospore is a dormant body that resists heating and drying. They have cell envelope structures such as a cell membrane and a cell wall. And that cell wall would determine if that organism is gram-positive, gram-negative, or acid-fast. And then they have surface polymers such as a capsule, flagella for motility, and pili and fimbriae for adhesion. Now, the eukaryotic cell structure would include cytoplasmic structures like a nucleus and histone-covered chromosomes, nucleolus, which is the site of ribosomal RNA transcription and processing. They also have rough and smooth endoplasmic reticulum, and that's going to transport proteins. They have mitochondria, which makes energy. They have lysozyme, which is an organelle that breaks down waste and debris. And they have peroxisomes, which are organelles containing enzymes that direct many metabolic activities like oxidation and lipid biosynthesis. They have cell envelope structures such as plasma membranes, cell walls, and motility organelles such as cilia and flagella as well. Giardia is an example of a eukaryote that has flagella. So you want to make sure that you take a good long look at the structure of a prokaryote because you may need to know that. Okay, so the prokaryote has a plasma and cellular membrane. The most inner membrane is the plasma membrane, and the outer membrane is the cell membrane. These are phospholipid and protein structures involved in the transport of molecules in and out of the cell. The plasma and cell membrane aids in energy metabolism and anchors that DNA during replication. The cytoplasm contains the internal structures, and the cytoplasm includes cytosol, water, salts, ions, and organic compounds. We have ribosomes, which is an RNA protein machine. The ribosomes actually assemble proteins. Okay, and then we have DNA, which is the chromosome that contains genetic material. And then we have pili for the attachment or transfer of genetic material to another cell. These are also known as adhesions. Okay, you may also hear of the conjugation pili or conjugation pilus, which is used for the exchange of genetic material. And then we have flagella, which is a means of propulsion or motility. Now, prokaryotes can live as single organisms, but a lot of times they are not necessarily living as single organisms, but instead they're living in environments known as biofilms. And a biofilm is actually beneficial to the organism because they can live in communities that kind of helps them live in a multicellular state. The organisms can work cooperatively for some benefit. So one benefit would be something like antibiotic resistance. That's one benefit of being or living in a biofilm. Let's talk about prokaryotic growth and nutrition. All right, so here we're going to discuss the reproduction of prokaryotes. The goal, or the ultimate goal of a prokaryote, is to increase the size of their population and to maintain an identical genotype of the parent cell. That's kind of the goal. Prokaryotes accomplish asexual reproduction without microtubular involvement seen in eukaryotes. Okay? Reproduction of prokaryotes is accomplished by binary fission. This is an asexual reproduction process, but first, before binary fission takes place, exact copies of the genetic material, which is DNA, must move to the opposite ends of the cell. This is called cytokinesis. So cytokinesis takes place when a ring of protein that forms around the middle of the cell cleaves the genetic material in the cell into two identical pieces. Okay? Binary fission is when the chromosome replicates and then separates. Cytokinesis is an inward pinching. This occurs around the two cells and pinches the two cells apart. The mother cell has now become two identical daughter cells. The time it takes for a cell to double binary fission really varies depending on the organism, okay? So E. coli duplicates relatively quickly. That's actually why it's a really good organism to study and do research on. It actually doubles in size logarithmically every 15 to 20 minutes. Then you have things like Mycobacterium tuberculosis that takes a lot longer to undergo binary fission, and their generation time is much slower. Okay, but when bacterial populations double at regular intervals, this is called exponential growth. It's used in infectious disease to determine the amount of time it takes for a disease symptom to develop. So a rapid generation time means that the person afflicted with that infection is going to have symptoms faster than in a person that has a longer incubation period. Now, all bacteria require three things for growth. They require a carbon source, a nitrogen source, and an energy source. A carbon source is either an organic source of carbon like a sugar or a fatty acid and iron, or some other form of carbon that is not organic, like carbon dioxide. Okay. You can have a nitrogen source for nutrition, and you have to have an energy source. The energy source for bacteria is ATP, and ATP, of course, is generated from glycolysis. ATP stands for adenosine triphosphate, and is the end product of phosphorylation. The bacterial growth curve occurs in 4 stages, and those stages are known as lag, log, stationary, and decline. So the lag is when proteins and enzymes are being made and are getting ready to multiply. The log phase is the exponential doubling of the population size, and it is steady binary fission. The stationary phase is when binary fission stops because the nutrients and space used by the organism has been exhausted. So fission stops during the stationary phase. The stationary phase is often referred to as the plateau stage. And then we have the death phase, or the decline phase, where viable cell population starts to die. All right, here are some examples of 2 different types of bacteria known as autotrophs and heterotrophs. Autotrophs make their own food by using photosynthesis, CO2, and water. All normal human flora, however, are heterotrophs. So autotrophs are organisms that use carbon dioxide as their carbon source. They oxidize inorganic compounds and they require water and inorganic salts to thrive. Algae and plants and certain types of moss are all examples of autotrophs. An autotrophic bacteria of importance is called cyanobacteria. And cyanobacteria contributes to the Earth's oxygen and plant chloroplast. Life cannot exist without cyanobacteria. The heterotrophs are things that require an organic source of carbon, so they require sugars and fatty acids such as glucose. They will oxidize or ferment organic substances, and all organisms that colonize the human body are heterotrophs. So all human normal flora and pathogens are considered heterotrophs. In order to achieve optimal prokaryotic growth, the environment has to be suited for them in temperature, gaseous atmosphere, and pH. Most human pathogens are mesophiles with an optimal growth temperature of about 35 to 37 degrees Celsius. Okay? Bacteria grow from close to freezing temperatures all the way up to nearly boiling temperatures, but most human pathogens are mesophiles. Gaseous composition in the atmosphere refers to how much oxygen or carbon dioxide is available to organisms. This affects the anaerobic respiration, anaerobic respiration, and oxidative phosphorylation, which is reducing phosphates to make ATP. And then finally, the pH has to be optimal for prokaryotic growth. Most bacteria prefer a neutral pH, but not all. Like I've said before, most human flora and human pathogens are mesophiles, and these are organisms that grow best at 20 to 40 degrees Celsius. The human pathogens are ones that grow at 35 to 37 degrees Celsius. Okay? Now, the psychrophiles are organisms that grow best at 10 to 20 degrees Celsius, and thermophiles are organisms that grow best at 50 to 60 degrees Celsius. Okay? Body temperature is 35 to 37 degrees, and most organisms are cold cultured at 35 degrees. Some pathogens, although considered mesophiles, can grow at lower temperatures than that of the human body. These organisms grow best at 37, but can be dormant at lower temperatures and still release their toxic substances. Staph aureus can cause food poisoning from its toxins being released in refrigerated foods. So, for example, old rice in your refrigerator that is contaminated with Staph aureus or even bacillus, the organism itself is dormant, but the toxins, enterotoxins A and D, can still be released even if that food has been refrigerated. We've talked about this a little bit before, so this should be kind of a review, but we have certain atmospheres that prokaryotes prefer. Different prokaryotes prefer different atmospheres. We've got our obligate aerobes, which are organisms that require oxygen for growth, and one such example is Pseudomonas aeruginosa. You will notice that Pseudomonas aeruginosa in the laboratory always grows at the very tippy top of a broth, and that's because it prefers oxygen or has to have oxygen. Then we have our aerotolerant anaerobes, which are organisms that can survive with oxygen but do not require it. We have our obligate anaerobes, which are organisms that cannot survive in the presence of oxygen. An obligate anaerobe would be Clostridium. We have our facultative anaerobes. These are organisms that can grow with or without oxygen. They prefer without oxygen, but they do fine aerobically, and that would be things like Staphylococcus. Then we've got our capnophiles, or our capnophilic organisms. These are organisms that require a little bit extra carbon dioxide. And some of our fastidious organisms that are capnophiles would be Streptococcus pneumoniae and Neisseria gonorrhea. And then finally, we have our microaerophilic organisms. These are organisms that require 85% nitrogen, 5% oxygen, and about 3 to 10% carbon dioxide. And one such example of a microaerophilic organism would be Campylobacter. We have to make sure that our pH is pretty neutral for prokaryotes. Most bacteria thrive at a neutral pH, and most bacteria live at a pH that is very neutral at 7.0 to 7.5, so it's a little bit alkaline. Human tissue has a pH of around 7.2 to 7.4. Okay? Organisms that can tolerate a lower pH are called the acidophiles. Okay? One such acidophile would be lactobacillus. Some streptococcus are also normal acidophiles as well. These are things that can be used in bioengineering to convert milk to buttermilk or dairy products to yogurt. Acidophiles that are human pathogens would be organisms like Vibrio cholera and Shigella, and that is the organism that causes cholera, and Shigella causes a really nasty type of food poisoning or tummy troubles that includes things like bloody stool. Okay, both organisms can cause human gastrointestinal diseases. Let's talk about prokaryotic cell energy and metabolism. So remember, the prokaryotes are the single-celled bacterial organisms having no mitochondria, no endoplasmic reticulum, no Golgi bodies, and no nuclear membrane. Okay, but we want to talk about prokaryotic cell energy and metabolism, especially for our class. Bacterial energy and metabolism are important to understand for identifying organisms and coming up with their bacterial resistance. Now, some prokaryotes or some bacteria do produce endospores. Okay, these are asexual spores that develop inside the bacterial wall, and they are developed in response to harsh environmental conditions. These are really resistant structures that resist chemicals, they resist temperature fluctuations, they resist high heat and drying out. That's what the endospore does. The vegetative cells are functional bacterial cells capable of binary fission, but the endospore will actually form inside of that vegetative cell. Okay, vegetative cells undergo binary fission while endospores develop inside those vegetative cells and will not undergo binary fission until conditions are ideal. Okay, an endospore is actually a dormant phase of bacterial life in which it will germinate when the time is right, becoming a new vegetative cell. In this picture here, you can see how the endospore appears as a bulbous area inside the vegetative cell. So if you look at that slide on the right, you can see those clear dots. Those are actually endospores inside of Bacillus or Clostridium species. And again, those spores are formed to resist heating and drying so that the vegetative cell can become more active when conditions are right for it to do so. Now, sometimes these spores are found on the very end. This is called a terminal spore. An example of a bug that has a terminal spore would be Clostridium tetani. That's the organism that causes tetanus. Now, Bacillus often has subterminal spores where the spore, I'm trying to say that, it's not easy to say. The spore is actually found in the middle of the vegetative cell. That is called a subterminal spore, and that can be seen in Bacillus species. Okay? Endospores are often noted more easily if you grow your organism out in a broth and then do a wet prep or stain it. Now we're going to start to talk a little bit about cellular chemistry. Okay, so there's a few terms you need to be familiar with. The sum of all biochemical processes taking place in a living cell is called metabolism. Anabolism is the synthesis or the making of large organic compounds. We're forming bonds which require energy. Organic compounds contain carbon, of course. Anabolism is when proteins, carbohydrates, and nucleic acids are made into flagella, cellular membranes, and things or structures like that. Catabolism is the hydrolysis of large organic compounds, the breaking of bonds that releases energy. So in catabolism, large molecules are broken down to smaller molecules to be used as energy for cellular operations. As a microbiologist, you do have to have an understanding of metabolic differences in bacteria in order to identify those organisms down to the species level. So organisms can be identified by determining the carbon source, looking at end products from the breakdown of substances, and looking at either alkaline or acidic byproducts of metabolism. And that'd be fermentation. Bacteria catabolize carbohydrates to make energy. Carbohydrates are just sugars. They do this using several biochemical pathways. Energy is produced by two methods, and that method is either called fermentation or oxidation. Okay? Now, we don't need to make these concepts too difficult. Fermentation is energy generation without oxygen. This is an anaerobic process, and you will see this happening in the butt of our tubes. Oxidation is when energy is generated with aerobic respiration or with oxygen. This is an aerobic process that happens in the slant of the tube, okay? So your glycolysis and associated pathways can be found in your Leboeuf textbook. So take a look at that when you have a moment. Remember the acronym OIL. Oxidation is loss of an electron or a loss of a hydrogen, but gain of an oxygen. And RIG is reduction is gain. So OIL is oxidation is loss, and RIG is reduction is gain. That's a good way to remember oxidation and fermentation. Of course, energy is required for metabolic reactions in bacteria to take place. Energy in bacteria occurs in the form of ATP, and ATP is the key energy source for bacterial cells and other living things. The most common pathway for bacterial sugar catabolism is through glycolysis, also known as the Embden-Meyerhof-Parnas pathway. This results in the conversion of sugar to pyruvate, generating ATP, and we need ATP for energy. Remember, bacteria requires three things: a source of nitrogen, a source of carbon, and a source of energy, which in most cases is ATP. So in heterotrophs, glucose is the starting place for bacterial fermentation and oxidation. Other sugars used as carbon source for bacteria must promptly be converted to glucose. Okay? So most bacteria need an organic source of carbon, and one such example of an organic source of carbon is glucose. Glucose is then broken down by fermentation to make pyruvic acid using three biochemical pathways. The Embden-Meyerhof-Parnas glycolytic pathway is also known as glycolysis. This is a variant of the glycolytic pathway used for sugar catabolism in pseudomonads. The pentose phosphate pathway is another fermentative pathway, and so is the Ender-Deuteroff pathway. Pyruvic acid is the key metabolic intermediate in fermentation. Bacteria will process pyruvic acid in a variety of ways, yielding different end products. These end products are what express phenotype. Okay? These end products are phenotypical markers that aid in the identification of bacteria. Okay? So if we take a look at fermentation and their end products, alcoholic fermentation results in ethanol. Okay? And then there's some other examples of fermentation and their end products. For energy extraction to take place, bacteria must undergo glycolysis and or fermentation. So the first stage of bacterial energy extraction is glycolysis and fermentation. Remember, fermentation is an anaerobic process. This is also known as anaerobic respiration. Many prokaryotes initially convert glucose to pyruvate using the Embden-Meyerhof-Parnas pathway. This pathway will disseminate organic compounds and provide energy for the organism. The Embden-Meyerhof pathway converts glucose to pyruvate, and then energy and nicotinamide adenine dinucleotide is released, NADH. Okay? This is an anaerobic process. Oxygen is not involved, nor will it affect glycolysis. The pathway can lead to many different end products when the pyruvate is reduced. Bacteria can be differentiated by these end products. Glycolysis occurs in 10 reactions involving 10 intermediates. The intermediates may also be useful for cell activity. Glycolysis is not just the generation of ATP. Take a few moments to look over this chart. You don't need to memorize this or anything, but it shows fermentation of organisms and their end products. So for example, lactic acid, lactic acid is the end product of things like lactobacillus and streptococcus species when they undergo fermentation. Okay? And then fermentation of glucose by Klebsiella and Enterobacter, the end product would be acetoin. So those are just some examples of the end products when undergoing fermentation. The pentose phosphate pathway is distinguished by the key cleavage of the enzyme phosphoketolase. Phosphokedolase cleaves the pentose phosphate into glyceraldehyde 3-phosphate and acetophosphate. This pathway is mainly used by lactobacillus and other organisms used in the fermentation industry or bioengineering when we're turning yogurt or when we're making yogurt or making cheese or winemaking. Only a few bacteria use the interdeuteroth pathway as a fermentation path. The interdeuteroth pathway is another catabolic fermentation pathway used rarely by bacteria. Many bacteria, however, especially Pseudomonads, use this pathway to degrade carbohydrates for respiratory metabolism. Glucose is catabolized to pyruvate using completely different enzymes than those used in the Embden-Meyer pathway or the pentose phosphate pathway. Respiration works in combination with glycolysis to completely oxidize substances. There are 4 additional metabolic components that are needed in respiration, including the Krebs cycle, an electron transport system, an outside electron acceptor, and ATP synthetase. The Krebs cycle is the most important pathway for oxidation in aerobic conditions. Pyruvate is oxidized and makes 2 ATPs. The Krebs cycle is an oxidative process in respiration by which pyruvate is completely decarboxylated to carbon dioxide. Decarboxylation is when an amino acid like pyruvate has a carboxyl group removed from that molecule. Carbon skeletons for biosynthetic reactions are created, and electrons are donated by pyruvate and passed through an electron transport chain used to generate energy in the form of ATP. This cycle results in acid byproducts and the evolution of carbon dioxide. This pathway yields 15 moles of ATP. Here's a nice diagram of the Krebs cycle and what's happening in the Krebs cycle. And you can actually find a diagram of this in your Leboeuf textbook. Here, the Krebs cycle is an oxidative process or a series of enzymatic steps in respiration where pyruvate is decarboxylated from glucose and results in the making of ATP. Pyruvate is converted to acetyl coenzyme A, CO2 is removed from pyruvate, and an electron is removed, reducing NAD to NADH. This happens before citrate is formed. So you have to have a basic understanding of glycolysis and the Krebs cycle. I'm not going to ask you for a bunch of details about glycolysis and the Krebs cycles, but just understand that glycolysis and the Krebs cycle are metabolic pathways that extract chemical energy from glucose to generate cellular energy in the form of ATP. All bacteria require carbon, nitrogen, and a source of energy, which of course is ATP. Bacteria must have the ability to use various sugars, carbohydrates for growth. At least the heterotrophs, anyway, need a source of organic carbon. Fermentation of sugars is detected by acid production determined by pH indicators. So we can actually see fermentation in a tube by the change in pH. So fermentation is when you have acids that are being created and it results in the color change of a tube. Bacteria will generally ferment glucose before other sugars, so glucose fermentation is not helpful in the identification of most organisms. The fermentation of lactose is helpful. Not all bacteria ferment lactose. Okay, so lactose fermentation is helpful, but not all bacteria ferment lactose. There are organisms that ferment lactose, but they have to have 2 enzymes, beta-galactoside permease and beta-galactosidase. Lactose is 1 molecule of glucose and 1 molecule of galactose linked together. So we can determine if an organism is a lactose fermenter or a slow lactose fermenter by the presence of these 2 enzymes. And we're going to discuss this more when we talk about the OMPG test for slow lactose fermenters when we get to the enteric unit. So this has been Unit 2, where we discuss microbial cellular biology, cell structure and function, and cellular activities.